This question is for Jay Lennon. So, you have shown some of the microbes for its longevity for 1,000 days. Have you tested anything with the spores or spore formers? How long that could survive in your test?
Yeah, so this came up earlier too. The spore formers are, I guess, largely restricted to the firmicutes. We did have one isolate, and I think there was no loss of viability in that particular strain over the 1,000-day period. So that was the most, that had the longest rate of persistence and lowest death rate.
And also you have tried so many different kind of, stains, like, to see the viability. Based on your experience, what would be the best one? Because all the three that people have tried, but they could not use that with, environmental samples because of the autofluorescence or non-specific binding. So in your experience, what are the best one that could differentiate dead from live?
Yeah, so for those people who aren't familiar with it, there's a whole suite of different dyes that people have used over the past 3 decades to kind of make inference about the metabolic activity of cells. I was just talking with John Hobby recently at the Marine Biological Laboratory, who started first using this, and I believe in the late 60s, using acridine orange. And there's some interesting properties I think that people stopped using that dye for, because you can actually infer not only inactive but active cells by the intensity of different colors that fluoresce with acridine orange. The 3 that we used just happened to be convenient because we co-stained all those samples together. So we needed signatures that would tell us live and dead. That was a cytox green. CTC, which is a dye that's taken up and reduced by cells that have an active electron transport chain, and by difference we inferred cells that didn't stain with cytox or CTC as having intact membranes, but not active, so we classified those as dormant.
So those are good for the known microbes in the microplasma, but when it comes to the environment.
Yeah, sure. I think that's a good point.
I'd like to thank you both for your terrific talks, and I have a question or a comment for Pieter. Pieter.
Thanks.
Dorrestein. You convinced me that in adults, the feet most represent the chemistry and microbes that are on the floor. And it would be very interesting, though, to study toddlers and find out how the chemistry on the walls and on the floor spread around their bodies and their mouths.
I can't agree more. I think it would be really wonderful to study infants that are crawling on the ground, that are licking everything, how that impacts the indoor chemical environment and their microbial communities. As a parent, I certainly would appreciate that. And I think there may even be real long-term implications to that. So I think Rob is talking about the constant re-inoculation of microbial communities. Maybe early development, that's an important process of establishing immune system. It would be just a hypothesis, but that's something that we can test. Hopefully the funding bodies will support that type of research.
So Peter, you bring up a really interesting point about us bringing in the chemistry as well as the microbes. And it's quite clear that we're bringing in, especially skin microbes, into all of our environments. And I'm just wondering if, if you knew ahead of time, say, so what the point is that if you put on the chemistry, that would affect your microbiology, say in your skin, like lathering on sunscreen, which would then affect potentially what you throw into the microbiology. Or if you switch, so have you done anything like, I tested someone and then they went out and I switched them and they put on all this DEET, asked how did that change their microbiome on their skin, and if that then goes into them, because that chemistry that we do to ourselves could have a really significant impact on what ends up in our microbiome. Are you looking at that effect?
Yeah, I think that's a great question. So we did a study where we took 12 people, 6 males and 6 females. it's not published yet, so we're finalizing it. But we basically, for the first 3 weeks, they weren't allowed to use personal care products. Second 3 weeks, very specific personal care products. And then third 3 weeks, they went back to their normal routine. And we sampled them, each of them weekly. There are some really fascinating results. One of those is during the personal care products, we saw a reduction in sex hormones, for example, that were being produced. But the microbial diversity, for example, shot up, at least in certain regions of the body. So we're now getting transcriptomics data. It's almost finalized for this. So we can also begin to figure out what are the activities that are actually changing on the skin. Now, we have not made the leap yet to look at that in the human habitat, but I think that would make a lot of sense as well.
So I remember on one of your slides, Peter, that you showed that you found some traces of medicines like antibiotics. In the chemical. Were those active? Were they still active, or were they just metabolites that were no longer able to?
These were intact molecules, so they presumably they're active.
Okay.
I mean, even at sub-MIC concentrations, antibiotics tend to still be active. They do something, so they will impact it.
But what influence would you think that may have in a enclosed built environment in creating more antibiotic-resistant bacteria?
That's a great question. They are probably directly related, but at the same time, no matter what environment you go into and you culture microbes from those environments, you will find antimicrobial resistant organisms. So it's a chicken of egg conundrum, which one came first? But I think we do have to think a little bit about those antibiotics that are left behind in the environment. Also, the antibiotics that are used in food. We started to look at, with Rob in the American Gut Project, and there we start to see some evidence of people that claim that they have never taken antibiotic for a year. We start to see evidence that they actually have antibiotics in their guts, and presumably from the food that they're taking. So it's something we have to consider and carefully study, I think.
I think it's very interesting to see the relationship between chemistry and bacteria. But from a technical point of view, we know that our view about chemistry is biased by the possibility of the molecules to ionize in the mass spectrum. So what is the bias that could possibly exist when we sample bacteria?
When we sample bacteria?
Yep.
From a chemical standpoint or from a sequencing standpoint?
From a sequencing standpoint.
I think that should be addressed to the NIST person. I think there are quite a lot of, I mean, every step along the way, there are some bias that you're introducing. How you prepare your sample, how you extract the DNA, to the whole informatic pipeline, as Rob was saying, you know, identifying platypus or dinosaurs in your fecal samples. There are biases along every step along the way, and so we need to come up with strategies that sort of create a uniform way of looking at how people do each one of these steps and come up with best community practices. And that's true for metabolomics as well.
Do you have any way to correct the bias that exists because of the ionization of the molecules?
So no, you have to run different programs. So you can use chemical ionization, then you can go and see, and then you can find molecules that do not ionize typically. And so there are ways of changing your workflow to begin ionizing the molecules that you otherwise would not see.
If I could ask a quick question to Jay. I'm just curious, how much have you looked at the potential, or is there the potential for genetic exchange between live and dead organisms in terms of possibly plasmids? Or so if you have that ability to make the separation, is that something that you've looked at or considered?
Yeah, so when a cell dies, it would presumably release some of its nucleic acids into the environment, and there's a lot of people who are interested in what the what the fate of that material is. In some cases, it can be taken up and used as a resource by other microorganisms. In some cases, it can be taken up intact through mechanisms of competency incorporated into the genome. So that's a mechanism of horizontal gene transfer that someone brought up earlier today. Yeah, and then of course, then there's just this issue of it perhaps biasing our estimates of microbial diversity as well. But I think there's been a long standing interest in the idea that the dead bacteria may be contributing to a pool of DNA that can be, I think Rosie Redfield called it genes for breakfast sort of idea that could be important for the evolution of bacterial populations.
I was wondering if you could say something about these two different technologies, sequencing technology and mass spec technology, to understand better and quantify the links between the human environment and the built environment. Right today, what would you say would be a better approach, using one or the other or both?
I'm a big advocate for both. So chemistry gives you different information. It doesn't give you chemical it doesn't give you taxonomy, for example. And so if you want to correlate it to taxonomy at this stage, because we don't have a reference database for metabolomics data of microbes with mass spec, you have to have the sequencing information. And so because without the taxonomy, you cannot really interpret the chemistry and how the roles that microbes play. So I think both of them are really important, in my opinion.
Yeah, those are great talks. And I'm reminded of a truism about biology and ecology, that we're much better at describing organisms than we are at characterizing the environments in which those organisms live and that they actually experience. And one thing that's really hard is it's not the average of the environment, it's sort of the variance of the environment and how they're structured in space and time. So dormancy only matters because there's temporal variability in the environment. And those beautiful graphs you had, Peter, showing that you have tremendous spatial heterogeneity in the chemical environment and the associated microbial communities just in the small confined space of your office. I think getting a handle on that is actually very important, because if you can understand diversity, you have to understand the environmental template that's maintaining that diversity, which means looking at variation in space and time.
So I think we can take one more question if we have one.
There's one last question up here.
Yeah, I had a question for Peter. A lot of the things you mentioned were touch interface surfaces. Did you look at any surfaces that weren't touched or interacted with, and were there any general patterns of chemistry there?
So, not directly. But I mean, the walls and the ceilings, people usually don't touch those. And so you can see that they have unique microbial communities, and I think it's pretty evident that people still dominate a lot of the microbes and the chemistries that come in. But if you don't touch it, that the contribution is diminished quite significantly. But it would be great to go into a place that has never been occupied by people and see what is a time-dependent change, in the same way that people have done it in the microbiome field, where they looked at a hospital a new hospital that just started, and then the transitions that took place as it started to be occupied. I'd love to do similar studies for chemistry as well. They just don't exist yet.
Speakers.